In Chapter 3, we build on our chemistry lessons from Chapter 2, and we apply what we know about chemical bonds and their interactions to our biologically important molecules. We're going to learn about carbon as the basis for biological molecules, and also learn about the structure and function of each of our four major types of biologically important molecules: carbon, lipids or fats, proteins, and nucleic acids. So in Chapter 3, if you've looked through the book, you'll notice that this is a really big chapter. Many of our chapters are really big chapters. And so I'm going to try to focus on the highlights, the big picture. I want you to understand the significance of carbon as a biological molecule, or as the basis for many of our biological molecules. I want you to know the difference in the structures, functions, and makeup of carbohydrates, lipids, proteins, and nucleic acids. So you'll want to know what they're made of, what they do, and why they're important. You'll need to know the 4 different levels of protein structure. This has to do with the building blocks of proteins. And then also get a basic understanding of nucleic acid structure and function. We're going to revisit nucleic acids, so we're really going to get just a super quick overview of those guys, but the other 3 we're going to spend a lot more time on. So, what do you need to know from this chapter? As I said, this is a really big chapter. Chapter 2 was a really big chapter. That makes Unit 1 a really big unit. You really need to understand carbon. You need to understand the bonds that it forms. You need to understand that its shape affects the way that it builds or is the basis for these other biological molecules. You need to be able to recognize the structures. I might ask you to draw a structure. I might ask you to label a structure. I might ask you to identify what an unknown molecule is based on its structure. So in order to do that, you're going to need to know the general characteristics of it. What do the monomers or the pieces of these molecules look like? What are the common atoms in this molecule or in any of these molecules? And what is their overall structure? How do they interact? How do the pieces interact to form the larger molecule? You'll need to know the dietary source of all of these. You probably know the dietary source of all of these, except for DNA. We really don't eat DNA, but we eat the other 3. And so you probably know where these come from already. You need to know the names of the pieces, and you need to know what all of these do in our body. Why are we even studying them? Why are they important? Because all of these molecules are going to come back to haunt us all the time, over and over and over in the remaining lectures in our semester. In Chapter 1, we talked about structure and function. We talked about emergent properties, where you get new functions as you organize parts and pieces. And structure and function and emergent properties is going to continue to be important in this chapter. So we talked about structure and function a little bit in Chapter 1. And structure and function is going to continue to be important when we learn about these shapes of our biological molecules, because the shapes of those molecules directly affect how they are used in our body. So all molecules have a unique structure, a unique size, and unique shape that are critical to their cellular functions. So when you are learning the functions of these molecules, pay attention to the structures, pay attention to the key components that make them up, because it's going to give you a hint about what they do in the body. Biological molecules in particular get their unique structures and functions because of carbon. And we're going to talk a lot about carbon, but just like oxygen from our last chapter, carbon has some really unique properties that make it important for biological molecules. So over here on the left, we have our water molecules, and water molecules form a specific shape because of the presence of oxygen there in the middle. That oxygen and the shape of the oxygen molecule, because of the hydrogens there, give it polar properties. So water molecules are polar. They're slightly negatively charged on one end, slightly positively charged on the other. And that allows for some really cool properties that is the basis of life on Earth. So carbon also has some unique properties, and we'll talk a little bit more about those properties in a minute. But just to get started, carbon has this really unique shape. It forms 4 bonds around a central carbon, and based on the way that those atoms around the carbon interact, you get a tetrahedron shape. So a tetrahedron is like a pyramid, and that allows you to have a very diverse molecule type forming around your carbon. So we can see that the structure of oxygen and water is really important. The structure of carbon and our molecules are going to be important in a few minutes when we start talking about it. But your book has this great example of why structure and function is important in biological molecules, using a molecule that is produced naturally in our bodies. So you guys have probably all heard of endorphins. These are our happy proteins, our happy hormones, our happy chemicals. They are released when we do things that make us happy. So when we eat, when we exercise, when we do things we love. They're also produced in stressful or painful situations. And on our cells, in our brain cells, we have these endorphin receptors. So these are protein receptors that are embedded in our membranes in our brain cells, and they have this little receptor pocket or location on the outside of the endorphin receptors that are shaped like this section of an endorphin molecule. And so endorphins will bind to those endorphin receptors, and they basically block pain. So that's how they make us feel good. They sort of block the pain from an actual painful or stressful situation. Now, morphine, you probably all know what morphine is. Morphine is an opiate. It's a drug that used to be prescribed as a painkiller. It's still prescribed as a painkiller sometimes, but the problem with morphine is that it's very addictive. And there are several other molecules like oxycodone that are similar to morphine. They're similar to endorphins in that they have the same shape on one side of their molecule that allows it to bind those endorphin receptors in place of your natural endorphins. So they mimic the function of your natural endorphins, which is why they can be used as a painkiller, but it's also why they're addictive, because you can take them to feel good. You don't have to wait for your body to produce them naturally. So here we see the structure of this one part of each of these molecules. The same structure is leading to the same function within our bodies. Now, I'm not saying you need to go out and find opiates. Please do not do that. But I'm just using this as an illustration of how important structure and function is in biological situations. So the rest of this video is going to focus on carbon. Carbon is the basis of all of our 4 major biological molecules. Besides water, living organisms are mostly carbon. And that's because all of those important molecules, carbohydrates, lipids, proteins, and DNA, are all made up mostly of carbon. So all of the major molecules in our body are mostly carbon. So why is carbon so incredibly important? Well, carbon is important because it's a structurally diverse molecule. That means that a lot of different things, a lot of different groups can bind to a single carbon molecule. And you'll notice that all of these biological molecules are essentially chains or repeating sections of carbon. Some of them are actually repeating carbons. Some of them are chains of structures that are made up of carbon. So let's talk about what carbon is doing at the molecular level. So I've said a couple of times that carbon has a really great structural diversity, and that is because carbon forms 4 bonds. Now, if you refer back to a periodic table, you'll see that carbon has 6 protons, which means in its uncharged state, it's going to have 6 electrons. So if we draw our carbon molecule, there's our nucleus and our electron shells, and fill them with electrons. So our first shell is going to have 2 electrons. Remember that 2 electrons always reside in the lowest electron shell. And then the outer shell is going to have 4 unpaired electrons for a total of 6 electrons in our carbon molecule. Now remember from chapter 2 that those electrons in the outer shell are the valence electrons. And those 4 valence electrons are each going to be involved in a bond. So around any single carbon, we can form 4 bonds, with something else. I'll just draw that back there. Usually, if nothing else is bound, there's going to be a hydrogen there. So this is our simplest carbon molecule. This is methane. It's a tetrahedron, which is our pyramid shape. And it is shaped like that because of bonding angles and the way that the electrons sort of repel each other. That's a little bit more chemistry than we're going to go into. But every carbon is going to have 4 bonds around it, or up to 4 bonds, in this tetrahedron shape. So you can imagine that there can be 4 different things coming off of a single carbon, and then the next carbon can have 4 different things coming off, and then the next one can have 4 different things coming off. Another thing that's special about these carbons is that you can form, or carbon will often form, double bonds. Very often with itself, sometimes with oxygen, but carbon will form double bonds. That means that there are 4 electrons being shared between these 2 carbons, and then each one of those carbons has 2 remaining bonds. And so you can, again, branch off those carbons to fill up those remaining bonds. So down here on the bottom, we have ethylene. This is our simplest molecule with a carbon-carbon double bond. This one, when you have the double bond, it actually makes a flat molecule. So now we've got a different shape that carbon can make. When it forms this double bond, it gets flat. So when it's got all 4 bonds, being all 4 single bonds around the carbon, you get these diverse pyramid-shaped structures. When you have the double bonds, you get a flat molecule. So if you have a chain and then you have a flat part of your chain of carbons, you've now changed the shape of that carbon molecule. The most basic carbon molecules are hydrocarbons. So remember that I said that carbon likes to form 4 bonds. So anytime it's not bound to something more exciting, it's bound to hydrogen. Hydrocarbon chains are basically carbon skeletons. So if you look up here, we've got ethane, we've got 2 carbons, and then they're saturated with hydrogen around it. Propane is 3, so this is just a chain of 3 carbons. These are our carbon skeletons. So they are just chains of hydrogens and carbons. You've probably heard the term hydrocarbon before. Hydrocarbons are important in our bodies as the basis for our fat molecules. But the reason for that is that the bonds between these carbon molecules here are really, really high energy. So you might recognize some of these names: propane, ethane, butane, butene. These are all flammable gases. So propane, you can buy canisters of this for like camping lanterns or camping stoves. Butane is the little, like the Zippo, there we go, the Zippo lighters. So these are basically flammable. And the reason is that there's a lot of energy in those carbon-carbon bonds. So this shows us one of the important functions of carbon, which is going to be energy storage in living organisms. So in living organisms, these carbon skeletons are often going to store energy. So you already know 2 energy storage molecules from your diet. They're going to be carbohydrates and lipids. This is why athletes will carbo load before a big race or a big intensive workout. They're storing up quick release energy. This is why our body stores excess energy as fat. It's storing this energy in these carbon-carbon bonds. The bonds between carbon molecules are non-polar bonds. Remember that any molecule any 2 atoms that are bound to one another are gonna form a non-polar bond, and that's because they have equal electronegativity. I don't have room for that there. Equal electronegativity, which means they're equally holding their electrons between the 2 molecules. So if you have a long branching hydrocarbon skeleton, it's going to be a whole bunch of repeating nonpolar bonds. So keep that in mind, because that is going to affect how some of these molecules interact or don't interact with water. So that's another important functional property of the structure of these carbon skeletons, is their ability to interact with water or other polar or other non-polar molecules. So the other place that you are familiar with hydrocarbons is in the form of petroleum. So petroleum is the basis for our gasoline. And that is a great illustration of the amount of energy that is stored in these carbon-carbon bonds. If you light any type of gasoline on fire, it just immediately goes up into flames, releases a huge amount of heat, a huge amount of energy. And that is because we've got energy stored in those carbon-carbon bonds. So before we move on to talking about our structures of our biological molecules, we need to talk about a few more properties of molecules in general. One of those properties is the ability to form different structures from the same molecular formula. So these are called isomers. So isomers are molecules that have the same molecular formula but different structures. So there are a couple of different types of isomers. One type of isomer is a structural isomer. In a structural isomer, we just have molecules arranged in a different fashion. Basically, a different arrangement of a chain. So on the left, we have pentane. Penta meaning 5, so we have 5 carbons here. And they're just in a straight line. Pentane is just a straight line of 5 carbons. On the right-hand side, we have 2-methylbutane. So I'm not going to go into why this is named the way it is, how it's named. There is a process for naming these molecules. But 2-methylbutane, if you count them up, still has 5 carbons. Both molecules have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 hydrogen. So both of these molecules have a formula of C5H12, but their structure gives them a different name and also different properties. Our next type of isomer is called a cis-trans isomer. So again, these molecules are going to have the same molecular formula, but they're going to have different arrangements of their atoms. So in a cis-trans isomer, we need a double bond. So we're talking a lot about carbon. So if we have a carbon-carbon double bond, we're talking about the arrangement of the groups attached to those carbons relative to the double bond. So if you look at the root words here, these are going to give you some information about what they mean. You might not be as familiar with cis, but the root word cis means same. So cis isomers are the version of the molecule where you have your functional groups on the same side of your carbon-carbon double bond. So here we've got our functional groups on the same side of the carbon-carbon double bond. So each of these X's here, this is just to indicate something is bound there. Each of those X's is attached to a carbon, but they're on opposite ends of the double bond, but they're on the same side. So in this image, it shows them on what we'll call the top side of the molecule. They're both on that top side. They're both on the same side of that double bond. Trans means different or opposite. So in a trans isomer, we still have that carbon-carbon double bond. We still have the functional group attached to each one of the carbons, but those those functional groups are on opposite sides of the carbon. So here we've got one on the quote-unquote top side and one on the bottom side. We'll talk about cis and trans again when we get to our fat molecules, and you're probably familiar with the term trans fat. We'll talk about what that means for a fat molecule in a few minutes, but it is related to the carbon-carbon double bond and the location of the functional groups that are bound to those carbons relative to the double bond. On the right-hand side here, we have a couple of examples, just simple examples. These are not necessarily biological examples, but this is 2 examples of a cis-trans bond. On the left-hand side, we've got our functional groups on one side, on the same or the cis side of our carbon-carbon double bond. So there's carbons there for a double bond. And then on the right-hand side, we have fumaric acid, where we've got our functional groups on opposite sides of our carbon-carbon double bond. The last type of enantiomer that we're going to go over, the last type of isomer that we're going to go over is called an enantiomer. So enantiomers are mirror images around an asymmetric carbon. So you know what a mirror image is by looking at your hand or anything in a mirror, but if you look at your hands, your left and right hand, no matter what you do, no matter how you turn your hand, you cannot superimpose them on one another in the same direction. The closest you can get is putting your hands together and getting a mirror image with your two thumbs touching, your pinkies touching, and so on. Molecules can do the same thing. So in an enantiomer, we have, in this example, a carbon in the middle. This is called an asymmetric carbon. And what that means is that it has 4 groups in different arrangements. So you can see our 4 different groups here. We've got carbons and oxygens up at the top. We've got hydrogens on the side. Notice they are on opposite sides. You can kind of think of them as on the thumb side. We've got carbon and hydrogen on the bottom, and we've got this nitrogen group on the sides, in the middle, kind of on the pinky side. No matter what you do, no matter how you twist and turn that molecule, you can never superimpose them on one another. They're always going to be mirror images of each other. Now, enantiomers and other isomers are really important to us because of their functional properties. So an example of an enantiomer, a biologically important example, is ibuprofen. So ibuprofen has 2 forms, an S and an R form, and only one of them really works. They have different biological effects on your system, and so when manufacturers are manufacturing ibuprofen, they have to be careful about which enantiomer they are giving you, and because it is not going to have the same effect depending on which one you are actually given. Before we get into our discussion on specific biological molecules, there's a couple of other things we need to talk about. We need to talk about chemical groups. So I've mentioned a couple of chemical groups without actually mentioning their names so far. Chemical groups are basically groups of molecules that are attached to the carbon skeleton. And they're important because they can change the property of molecules, give them unique properties, give them different properties. So on the bottom here, we have a steroid. Steroids are basically hormones in our bodies. And if you look at the overall framework of these 2 molecules, they look really, really similar. This is estradiol and testosterone. Estradiol is a hormone that is important in the female body, testosterone in the male body. But the difference in functional groups around these molecules are going to give them different properties because they are going to behave differently within the body. So each one of these is a particular type of chemical group. They all have their own properties, and that's going to affect how they behave. There are several common functional groups. These functional groups is just another name for a chemical group. Functional groups are important because they are going to be involved in the functional uniqueness of a molecule, but they're also potentially going to be involved in chemical reactions in a very specific way. They are often going to affect the shape of a molecule. They're going to affect they're going to affect how that particular molecule or region of that molecule behaves with other molecules or with other parts of the molecule. So there are a few chemical groups that are really important. They're going to keep coming up, especially today as we go through chapter 3. I would strongly encourage you to memorize these because they are going to be important. I'm going to talk about these over and over again. You're going to want to know their general properties. Just to point out a couple, a hydroxyl group, we're going to see that a lot. This is an OH attached to anything. So our example here is ethanol. Usually, if it has an OH on the end, it's going to be an alcohol of some sort. So you've probably heard of ethanol. You've probably heard about ethanol. We'll use it in our labs a couple of times. Ethanol is basically a methane. We saw methane earlier. It's basically a methane, a 2-carbon structure with an OH on the outside. Carbonyl and carboxyl groups are going to be important. These guys are important because of the oxygens, and same thing with the hydroxyl group, because they are polar, because they like to give up their hydrogens. That's going to make them acidic. We've also got amino groups or amines. So these are important in proteins. Every single one of our protein monomers, or the pieces that make up proteins, are going to have an amine group in them. This one here, glycine, is a single amino acid, hence the name amine. Those amino acids are going to be our basis for our proteins. Phosphate groups can be really important. In just a minute and in later chapters. Phosphate groups have a lot of energy. They're very unstable, and so they are going to be important in our energy molecule, adenosine triphosphate, or ATP. You might be familiar with ATP from other biology courses. So I would suggest that you learn these names because I'm going to say them, and while I am not specifically going to test on them, it will make understanding some of these molecules a little bit easier. So let's talk about our adenosine triphosphate really quickly. It's not one of our big biological, let me rephrase that. It's not one of our big, large biological molecules, but it is one of the most important biological molecules. And the reason for it is that it's got a series of phosphate groups. So these are high energy, they are very reactive and very unstable. And ATP contains a lot of energy in these unstable oxygen and phosphate bonds. And so ATP is essentially the energy currency in our cells. It's the molecule, the main molecule that stores and transfers energy within our cells. So if we look over here on the right hand side, we've got a big picture of the ATP molecule. Basis is carbon. It's actually very similar to a nucleotide. We'll see nucleotides when we get into DNA, but both of our pieces here of our ATP are carbon based. We've got these carbon nitrogen rings, and then we've got a carbon ring structure, a sugar. So we're made up of a sugar, a nitrogenous base. That's the name for this guy up here. And then our triphosphate group. So we'll see this general nucleotide structure again when we look at DNA, but keep this structure in mind, this triphosphate, adenosine triphosphate, because we're going to spend a lot of time talking about this guy when we get to cellular respiration and photosynthesis. Energy is stored in this molecule in the phosphate groups. When one of those phosphate groups is removed, when it's kicked off, because this whole triphosphate section is really unstable, it's going to release energy. And that energy is what powers our cells. That's how we store and use energy. We're not using electricity, we're using ATP to run our cells. So we're about to get into talking about our carbohydrates, lipids, and proteins. as a general overview, these four biologically important molecules are called macromolecules, meaning large molecules. They're really big molecules. And the reason that they are macromolecules is that they are polymers. So poly meaning many. They are repeating units of many molecules. So we're going to be stringing together individual carbon and carbon molecules to get these large macromolecules that we call polymers. So of our four biologically important molecules, carbohydrates, proteins, and nucleic acids are polymers. This means they are made up of units that are covalently bound together. So polymers are long molecules consisting of many similar or identical building blocks. Monomers, meaning one, mono, meaning one, are the individual building blocks. So you'll notice with these three molecules, they are just repeats of the same thing over and over again, or repeats of very, very similar things over and over again. Most of the metabolic processes in our body have to do with breaking down and building these polymers. So when you eat, let's say you put some sugar in your coffee, you consume that sugar. Your body has to break it down because it's going to use the individual molecules in that table sugar to build other things. So you're going to break down the food that you eat. And that means at the cellular level that you're disrupting the bonds, breaking apart the larger molecules into pieces that your body can use. This happens because of enzymes. So enzymes are the most important type of protein in our body. They are special proteins that work to mediate chemical reactions. They make the chemical reactions in our bodies go, including the breakdown and the building of these big macromolecules. So really quickly, we're going to talk about the ways that we build and break these molecules. And we do this by two different types of reactions. One is a dehydration reaction because it releases water in the reaction. And the other is hydrolysis. Hydro meaning water, lysis meaning breaking. It's going to use water in the reaction. So we build polymers through a dehydration reaction. So in a dehydration reaction, we are going to combine a hydroxyl group and a hydrogen, two H's and an O. We're going to pull those out of our long polymer that we're adding onto and the new monomer that we're linking. We're going to release water in the process. So we're going to basically dehydrate these two starting molecules. We're going to dehydrate them. We're going to release that water. And by doing that, we can link those molecules together into a long polymer. Alternatively, we can break down polymers. We can break down polymers by the process of hydrolysis. It's basically the backwards reaction of a dehydration. So if we want to break this bond here between monomers 3 and 4, we need some water. We need to put back that water that we took out when we built it. So we're going to split this water. That's the hydrolysis. Split the water. Add one hydrogen to one end, a hydroxyl group to the other, and now we have released that monomer number 4 there. So all of these molecules, all four of them, are going to be built by dehydration reactions. Water is going to be released when we put the pieces together, and they're going to be broken down by hydrolysis. We're going to take water in, we're going to split it, and we're going to use it to remove the monomers one at a time.